Influenza A. Influenza A virus is highly contagious and can spread through tiny droplets of bodily fluid from coughing, sneezing, or talking. Someone may even catch the flu by touching their mouth or nose after coming into contact with a surface or object that has a virus on it. Influenza A is caused by an infection with a virus. It is often called the flu. There are 3 types of influenza virus, A, B, and C. Influenza A is more serious than B and C. It is the only type known to cause widespread outbreaks. So, within this model, we'll be covering influenza A. So, the symptoms of influenza, according to this diagram right here, would be central headache, systematic fever, muscular tiredness, joint aches, nasopharynx, runny or stuffy nose, sore throat, aches, respiratory, coughing, gastric, vomiting. Okay, so the third line of defense, before we get into the whole respiratory system within influenza A, I'll explain the third line of defense briefly and its adaptive response, and then explain how influenza A travels through the human body and how the immune response reacts to this. Okay, so first we're going to cover search, remember, and destroy. But before that, I'm going to explain what a lymphocyte is. So a lymphocyte will be the main cell that we'll be talking about within the third line of defense. It's pretty much what makes up the third line of defense and what stops antigens from affecting your body and what releases them out of your body. So lymphocytes make up your third line of defense. They're a special white blood cell due to their specific defense mechanism. So this pretty much means that their specific defense mechanism means that they target certain antigens, specifically that antigen, and in these steps, they search, remember, and destroy. So lymphocytes live in lymph nodes, but also in the bloodstream and all over the body. Lymphocytes come in 2 main types, B cells and T cells. So within this diagram, we have lymphocyte X, that will be seen right here, and we have antigen X. So antigen X we could just call the flu. In our case, the flu. So flu, influenza A. So flu A. So within the first step, we'll be searching. So the lymphocyte will be searching. So the T cell lymphocyte will be searching for the antigen. So antigens trigger T or B lymphocytes. So the lymphocyte will be triggered by the antigen. And the lymphocyte will then remember the antigen and it will clone the antigen. So that can be seen here in this diagram. So the clonal selection, this will then clone. So after cloning, that will then further on destroy the antigen and remove it from the body. Onto the adaptive response. The correct adaptive response would be cell-mediated in influenza A. So how the adaptive response briefly before I continue. So right here we have the T cell, which looking back here will be our lymphocyte. So this will be our T cell lymphocyte, and this will be our antigen, so the flu. And a diagram here, we have it here as well to demonstrate. T cell lymphocyte right here. This will be our antigen, which will be the flu, and these are all the clones of the T cell lymphocyte. So T cells recognize antigens with their antigen receptor, which can be seen right here. A complex of 2 protein chains on the surface. They do not recognize self-antigens, however, but only process antigen presented on their surfaces in a binding groove of a major histocompatibility complex molecule. So further on, pretty much what cell-mediated is, it's direct, and what it does is it finds the antigen, and it is pretty much these T cells are designed to take out the antigen. So this will be matching up with the shape once that is made of the shape, as we go back here, it'll remember, it'll clone it, and it will destroy it. So that will be the process of it. T cells can detect the presence of an intracellular pathogen because infected cells display on their surface peptide fragments derived from the pathogen's proteins. These foreign peptides are delivered to the cell surface by specialized host cell glycoproteins. So moving on to the actual process and what happens within the human body in the immune response and how that cell-mediated immunity responds, so the adaptive response. So a few things that we have to make sure that we know, or a few things to go over, is that its genome is single-stranded negative RNA. Its genome is also segmented, so there are 8 strands of negative RNA. This can be seen right here as well. It is also enveloped with 2 key proteins in the envelope, hemagglutinin and neuraminidase. So that's right here. So like we were talking about before, you were initially infected with the Influenza A virus via respiratory droplets. So as we can see here, we talked about how the bodily fluid can contract through your body through coughing, sneezing, or talking. So that is pretty much through respiratory droplets. So onto the diagram right here. The droplets will then infect the epithelial cells. So this is your epithelial cell right here, this little box, in the respiratory tract. This happens because the hemagglutinin binds with the sialic acids on our respiratory epithelial cells. So what this is saying here is this is the droplets. This will be the Influenza A droplets. These then bind with our sialic acids. So the hemagglutinin will bind with them. As you can see the color here of this, all the way down, will bind with it. Once it binds sialic acid, it is then taken into the cell by the receptor-mediated endocytosis. So that will be seen right here. As we can see, it is now taken into the cell, and then this is then put into the endosome of the epithelial cell. Okay. Then H plus ions, hydrogen ions, enter the endosome, and that causes the envelope to fuse the endosome membrane and release its contents into the cytoplasm. So as we can see right here, H plus ions enter the endosome, and then that releases it into the membrane. So down here, as we can see here, into the cytoplasm, sorry. Next, the genetic material goes into the nucleus. Due to no human enzyme being able to make a negative strand, as we can see back here, it was a negative strand, and no human enzyme is actually able to make a positive strand from a negative strand. The virus brings its own RNA-dependent mRNA polymerase into the process. So once the virus has been made a positive RNA strand, those are then translated into proteins. They then bud off into the cell membrane, right up here, where the neuraminidase will then chop off their sialic acid, preventing the virus binding with them again. So pretty much what this is doing is the neuraminidase is chopping off the sialic acid, as the process would repeat, and the virus would not leave the body. So neuraminidase actually chops off the sialic acid from bonding with that again, from bonding with the virus, and it releases the virus from the body. So as you can see here, this is the virus, and that is the process and the immune response to Influenza A. So a few things we can see here in this diagram. The virus enters the respiratory tract. So pretty much this is the whole thing, and this is how it's fought off. How the immune response reacts. So memory formation from response. So currently the best protection against IAV is annual immunization with trivalent or quadrivalent vaccine, which induces neutralizing ABS to the viral surface glycoproteins, which would be hemagglutinin and neuraminidase. However, as these glycoproteins mutate frequently, it is necessary to reformulate and administer vaccines annually to maintain protective immunity. So, in conclusion, to the memory formation from this response is capable of producing memory T cells, although it is irrelevant due to the constant mutation in influenza that vaccines provide the only protection from it. So pretty much this process can produce memory T cells, but although how much influenza will how much it mutates, it's not really necessary, as a vaccine has to be made every year to prevent this. So there's not really any memory T cells that are necessary within it. So limitations in the model were, since the model was only a simplified version of the real objects, it does not work or function exactly like the real ones. It is only the best type of model to represent a very complex system because it cannot account for all the variables that influence or affect such system. Things such as H plus ions, hydrogen ions cannot be implemented into my model based on a symbolized object due to some objects being too complex to fit. And that's it.